Thank you everybody for coming to the second part of the morning. To begin, I would like to thank our Diamond sponsors and all the sponsors for the conference. Please go visit them at the sponsors section. You will find some amazing sponsors there. You will be able to have wonderful conversations with them, and they will be able to solve all of your problems. And to get things started so that we continue with the flow of this conference that has been amazingly on time, I would like to welcome our first speaker today, Rosaura Magen Estrada. So please, Rosaura, come join me in the podium. And we will listen to her first, and then we'll continue with this amazing session. Welcome.
Okay. Thank you, Mariana. So, I'm Rosaura. I'm from Mexico. And I would like to talk about a slides collection in Mexico City. This is a talk trying to explain to you the first steps to do this collection to be an usual and practical collection to visit in Mexico. So we have about I don't know, maybe 30 years ago, we found the collection and we start to revise. But there is a lot of problems. There are a lot of problems to do this. So, constructing a ciliate slides collection in Mexico, first steps. The protozoology laboratory belongs to a comparative biology department in Faculty of Science in a National Autonomous University of Mexico in Mexico City. So this laboratory was founded about the decade of the 60s decade. This since this time, the work was devoted mainly to taxonomical identification of protists in this case, and ciliates is my main group of protists that I have been working. But unfortunately, the slides were lost because the researchers did not have the opportunity to construct a collection. So we have different stains and impregnation techniques during the period of the 60s until 1980 decade, and all these slides were lost. The researchers usually conserve for their own purpose, and some others were maybe, I don't know, they tried to keep, but others were lost. So we found several papers where the ciliate species were recorded, but no slides collections. So, because these microscope slides were not deposited in any collection in all the country, we found in my laboratory these some slides about I don't know since the 19th decade, and all they are the result of projects, research projects, but also theses and so on. So we aim to check the condition and quality of the slides to formalize an institutional collection in the medium term. So you can see here some boxes with the slides inside, obviously. And these collections contains the data about 90 ciliate genera, including free-living species and symbionts. So this is a long, long list of genera of ciliates. And in here are the names of the Mexican states where these genera were found. So the genera, of course, for example, Acineta, as you can see, is a genera very common, not just in Mexico, it's around all the world. And it's a very important species because it's a kind of a genera of with a stalk, and the shape is maybe not so difficult to maintain. So this is very important record for all the protistologists. So here is the Mexican Republic map. You can see here. No records, white, and these colors indicates the number of genera were distributed in all the states. So we don't have genera in ciliate genera specimens in our laboratory. No slides, no information. There are researchers, yes, but the slides are not present. So the important here is to see the number of genera that we can obtain information to preserve these slides in a good condition. Some examples of ciliate, protist ciliates. These Bromeliotrix, for example, and also Facodinium. So there are species that lives in bromeliad water tank, very special habitat. Coturnia, for example, these are symbionts of the gills of several crustacean species. So each genera contains a species very important to not only to know the biodiversity of ciliates, but also the conditions of the habitat, tropical, forest, crustaceans, and so on. So because of this, we can try to maintain this collection in with conditions, to try to share with all the protistologists, okay? So our goal is to inform and share the challenges faced to maintain this collection and keep its institutional recognition. In Mexico, we don't have non collection of this kind of slides. So we found different types of slides. For example, species types. You can see here paratype. Yes, Coterilia bromelicola was found in Tillandsia heterophylla, and we can see here also in Coterilia, and so on. So because for example, the bromeliads have special conditions, water special conditions. The record of these species are not so frequent because we have to wait the rainy season to obtain a amount of water to recognize these species and so on. So also we have slides from thesis results. I am undergraduate, master, and PhD students. The problem with this is to revise each slide to check if the label is with the original data and to check if the ciliate has a good condition from staining and so on. There are a lot of boxes with slides. So some genera have been published in some papers, and this is important because if any person have the intention to revise, the collection is in there. Also, we have problems with the conditions to preserve. Yes, the label, the environmental condition, and so on. But also, we don't have a special spaces to maintain these boxes. So all these are the challenges for us, and I think it's very important to do the effort to maintain this collection in a proper condition, because, for example, some water, I don't know, maybe lagoons and ponds, they no exist anymore. So this is the big problem for us. But I would like to thank to Carlos. He's the technician, and of course we are two, only two, to do this work. Thank you so much for your attention.
We have time for one question. All right. If you are still thinking about a question and you have not yet come up with a question, we will have time at the end of this session for discussion. So I encourage you to start thinking about those questions, and if we don't have time for them, come back at the end. We have 1 little minute for transition now. So in the meantime, while we do that transition, I will still like to call Stevie Kennedy Gold to the podium. Start getting ready, warm up those muscles. There we go. Let people come into the room. There we go. We are slides in a slide box. This is very cozy, very intimate. Well, not too many, because, but yes, we are very much on time and very happy. So, all right. Yeah. Okay, I will give you a sign.
Okay. Sounds good. Awesome. Hi, everyone. Thank you for coming. My name is Stevie Kennedy Gold. I am the Curatorial Associate of Herpetology at Harvard University's Museum of Comparative Zoology, or the MCZ. Today, we're taking it one slide at a time and establishing a workflow for accessioning large donations of histological microslides. Brief outline, basically the bread and butter is we broke this process up into the physical side and the digital side. And because we're on a tight schedule, let's just jump right into it. We're all here, we probably know what histology is, but just in case, And so we all have the same background. Histology is the study of microscopic anatomy of cells or tissues, protists, as we just saw as well, but using a microscope. So on this giant picture here, it's actually about 1 inch by 3 inches, and that stained material is the tissue or the element of interest. So at the MCZ, we have a fairly robust collection that encompasses both invertebrates as well as vertebrates. And two notable vertebrate collections are those of Charles Minot and R. Glenn Northcutt. Together, they were hugely embryological in focus, and across the board, they represented numerous taxa, so mammals, herps, you name it, and they represented approximately 3,400 individuals over 63,000 histology slides, and those are still getting worked up. But I want to dive a little bit further in and focus on another researcher completely, Dr. James R. Stewart. So he is a herpetologist, recently retired from East Tennessee State University. He was interested in reproductive physiology and specifically the evolution of the amniotic egg in viviparity or live birth. He called us up last year and said, "Hey, I want to donate my life work to you. Can I do that?" We were like, "Yeah, sure, how much?" And he's like, "Oh, maybe 55,000 histology slides." And we're like, "Yeah, okay, we got that. Let's do this thing." So he drove up one man all the way from Tennessee to Massachusetts in, again, one minivan, about 1,500 kilometers. We're like, "Yeah, okay, 55 histology slides. What's that even look like in a minivan?" It was 61 cardboard boxes. Each of those boxes had about 9 histology slide boxes. So we had a total of about 549 histo boxes, each containing about 100 slides each. Conservative estimate was 55,000 slides. It turns out he kind of overpacked some of those cardboard boxes, and we had more towards 63,000 histology slides from one man's life work. So we had our work cut out, and it was overwhelming at first, so we split it up. We first tackled and processed the physical element. And of course, that involved unpacking the minivan, which was a lot. We were aware that critters might be present, so we put all of our boxes into a little quarantine area where we unpacked them from those cardboard boxes. You with eagle eyes might see we've started placing them into these more open air plastic containers. And if we zoom in even further, we're conducting inventorying as we unpacked it. So that's what these little labels are. In this video, you can see that I'm comparing one of the many printed Excel documents that he provided to the histology slide boxes and double checking the contents of the box and seeing the positions of the slides as we worked. It was laborious, but it was worth it. And I'm happy to report too that as we worked during this quarantine session, we were working with pest management. We did not want critters to infiltrate the rest of the collections. So we had that barrier of sticky traps at the bottom there. Happy to report, nobody crawled out. No, nobody was caught, thank God, but we really wanted to make sure. So we did put the slides in those open air containers into a CO2 or an anoxic chamber for about 3 weeks. At that point, we were comfortable and confident and brought not only the slides, but as you can see, the notebooks that he provided with all the data. We put all that in the bubble, and then we could bring it confidently all into the collection. And that's where we started to barcode them. So you see right here, these are the barcodes that we used. They are itty bitty, about 1 centimeter by 1.5 centimeters. In each of those barcodes is a unique identifier. So my intern meticulously placed each barcode on each microslide for use later. And so you're aware, if you're curious, we purchased our barcodes from electronic imaging materials, about 10,000 barcodes to one roll. So we had a lovely tight box of itty bitty barcodes. They're great. The next stage is to rehouse it eventually, because 63,000 slides takes a lot of time. We're still working through the barcoding process, and we're also waiting on our cabinets because they take time too. So in the meantime, and I encourage this as well, microslide cabinets are like top tier. That is what I hope everyone can have for Christmas or Hanukkah or whatever you celebrate. But in the meantime, maintaining them in their original plastic containers is good as long as you maintain it so that the cover slip is on top and the microslide is on the bottom, and you have a nice little gravity sandwich. Let gravity do the work for you and keep it all together. So this is, maybe next year's Spinach talk, I'll talk about how to house everything like this, but stay tuned. To switch gears, we also had to process the digital side of things. And this is because not only did he provide us his slides, as I referenced earlier, he also provided his preparation notebooks. And these are notebooks outlining the collection and the euthanization of the mother specimen, the removal of the egg and the embryo from that mother, and then the staining, the embedding, the slicing, and the placing of that embryo onto a microslide. So it was a lot of data. we wanted to make sure we had a digital archive. So we took all those notebooks, we scanned them, we individually saved and renamed each of those pages, and we proceeded to transcribe that. We were also working through this because, again, we had about 7 notebooks. It's a lot of data. And eventually, again, work in progress, we'll be connecting those scans and that data to the specimen records to try to make as holistic of a specimen record as possible. In addition, he provided us multiple spreadsheets, and we made our own, shocking. But they broke the data and the quantities down in each of the boxes and for each of the specimens. And we had rows on rows on rows of data that we had to conglomerate into this massive data set, and we're still working through that. But ultimately, that data showed us that we had, as I said, 62,375 microslides created from 919 individual specimens or specimen records, representing 33 different reptilian species. So if you remember, I mentioned the Minot and the Northcutt collections spanning multiple taxa, so multiple departments were invested in those collections. This is just herpetological, so we did have support and help, thank goodness, but it predominantly resided in the herpetology department. So we realized, holy moly, we have a lot of data. We have notebooks, we have Excel sheets, we have the information on the slides, we have information we're making ourselves. What do we do with this? Databasing is going to be a beast. And it's also important to recognize that the entire slide series, again, represents a single individual, a single sectioned embryo, which represents one specimen record in our museum. It was a lot to wrap our brains around, so it was easier to break it down. We use this inverted triangle regime here. Take a step back. One egg, one embryo is one specimen record. That's why we have 919 of both. This is a representative of our database system, MCZ base. It's applicable everywhere else. It could be a line in an Excel spreadsheet. That's totally fine too. If we dive further into this database, you'll notice that we have this section called parts, and those are the specimen parts, and that is the microslide series. That's important to note because in our database system, we can break down our parts into individual parts, which are those individual microslides. The series for Dr. Stewart's collection ranged anywhere from 21 to 164 slides per series, so there's a lot of variability. In this image here, there's actually only 8 slides represented. Imagine, 164 of them. It's going to be a very robust specimen record. And we introduced more information to those parts, as you can see here. So in this column at the top, it says container name, and then it's just a garble of numbers and letters. Those are actually the barcodes, so that unique identifier that I referred to earlier. It will help us later with locating the specimen or the microslide when we eventually put them into the cabinets. If you zoom in here, we also have attribute data for each of those microslides, and we glean that information from those notebooks, from the Excel data sheets. And it provides information like the section interval, the stains that were used, the thickness, maybe the date of preparation. So there's a lot of data mining that we're working through too. So to summarize this crazy complex process, again, we broke it down, the physical side and the digital side. We had to unpack, inventory, manage for pests, none present, barcode, and eventually rehouse. When it came to the digital side, we had to combine all of that information from the notebooks, the spreadsheets, the stuff we created ourselves into one specimen record, or 919 specimen records. There's a lot of work to do. There's a lot of headaches and a lot of questionings and sitting there scratching our brains, but I want to provide you with some takeaways that we realized during the entire process. So first, and I think this goes for all of our jobs, just anticipate and accept the delays. They're going to happen, snags happen, it's okay. Make contingency plans. So for instance, we're waiting on the cabinet to still come, so we hit transcribing really hard. You don't know how long barcodes will sit in the mail, so we started scanning extensively as well. Always make sure you make protocols and take notes. That's because you can then refer back to it and keep a standardized process throughout the entire endeavor and not scratch your head and say, what did I do 5 months ago? Oh, I don't remember. Or if you have a change in the guards and new interns or helpers come in, everyone can keep consistent. Second, again, this applies to everything. Demand the data and the permits up front. I recommend this across the board. Don't accept anything if you don't have that information because, again, saves you a whole heck of a lot of headaches. Third, and this was information from my graduate degree actually, is know when to ask for help and don't be afraid to do so. We all ultimately want to be in this top corner here. We love to share knowledge. That's why we're at this conference. And everybody here has their own unique backgrounds and information. They have the knowledge, ask them for it and share it. We all end up happier that way. And finally, be patient and have fun doing it. This is Dr. Stewart's life work. we treated it with respect because holy moly, we're learning a lot from it. So have fun with it, but be patient. With that, I ultimately want to thank all of you for sitting tight, like nice little sardines or slides in a microslide case, and as well as all my co-authors, my coworkers, my volunteers, and interns that helped make this project happen and keep happening. So feel free to contact me, and happy to take questions if there's time. So thank you.
Don't escape, we do have time for questions. So any questions, please go to the microphone. Do you have any online questions at the time? Not
Nope,
right now?
there's none.
Okay, thank you.
These roles of pre-prepare barcodes. I suppose you buy the barcodes which are LSIDs or unique or something like that. Do they have any advantage about printing your own series of barcodes?
I'm sure you could do either. So across the MCZ, our museum, the barcode numbers are actually used across multiple departments and across multiple preparation types as well. So I know they're used for microslides tissues, our tissues as well. Thanks, Cynthia. But you can print your own if necessary as well. So that
Yes,
I've seen that done absolutely. I know there are really intense systems to do that. However, you can, I think using your own printer, I've seen people just do like what are they called, zebra barcodes?
Yes,
Using those
that's
as well.
the usual practice. But I wonder, these barcodes that you buy, they are guaranteed to be absolutely unique. I mean, they are kind of
Yes.
long
Yes.
QII.
Yeah, so on that image, I apologize for that too. Thank you for clarifying that. There's only about 4 digits on that specific barcode, but it's a whole slew of numbers that we don't see because they're so small. But they are unique, yes.
And you find this really advantageous over having your own?
Yes.
Because I suppose, machine readable rather than human readable.
Yeah, there is that element, yeah. So there is the aspect too that you need a barcode scanner enabled to read them. But it does prevent any, typos and confusions in that
Any mix-up.
element. Exactly. And you can barcode scan when you place on the slide, when you move the slide into a cabinet. So many, paleontological, for example, I saw a talk earlier today, their entire storage space has a barcode that they scan and then the slide or the element. So
thank you.
it helps. Yeah.
Thank you.
Just to tack onto that, the pre-printed barcode. Just to tack on, I'm co-author on the talk. So the pre-printed barcodes are, they have the adhesive on the back of the barcode is meant to be resistant to a variety of solvents. So for cleaning the slides, if we're cleaning with distilled water or whatever, they're not gonna get wet like they might if you just printed your own.
Yes, but
Makes sense.
Printers that can use vinyl times, basically it's more thick before. Anyway, get your point. Thank you.
We have a question online? Can you read it?
How do you deal with parts of Dr. Stewart's notes that are in bad handwriting?
How do you deal with parts of Dr. Stewart's notes that are in bad handwriting?
Yeah. Isn't that the case that we all have with everything, right? There are elements that he actually was very meticulous in every capacity. I'm very grateful for that. There was a lot of cross-referencing. So as we went through, we're like, does that say benzene? And we kind of did a lot of matching up together. There was a lot of that element too. So it is contextual in many capacities. There's also a lot of Googling because I don't do histology. My intern didn't do histology. And we're like, is this a chemical used in this? Oh, it is. Okay, great, cool. So there is that aspect. I would also recommend just reaching out to other people too, if you're not sure, because a lot of times these researchers donate to other museums as well. So I know the mother specimen was at other places. So maybe the data was there as well. So definitely reach out, ask questions if you have them. So, thank you.
Okay, thank you very much. I do have a question, but I will save it for the end. I would like to invite now the Yale portion of this event. So I would like to invite Gregory Watkins Colwell, my co-author. Hi, I don't know you. President-elect of Spinach also. Anyway, thank you, Greg.
All right, so I'm the first of 3 talks about the same collection of slides. I'm sort of the part of the trilogy with the history and the origin story. So I'm like the Fellowship of the Ring, and then we have the Two Towers and the Return of the King later. Ice fishing cover slips. So my job is to tell you what's an ice fish. Antarctica is a continent. Antarctica is cold and it is hard to get to. It is surrounded by what I learned were, those oceans, all right? But now there's this Southern Ocean that they refer to around Antarctica with some really cool seas and ice shelves and things. It has always been hard to get to. In 1916, Shackleton put this really cool ad in the paper for helpers. Men wanted for hazardous journeys, small wages, bitter cold, long months of complete darkness, constant danger, safe return doubtful, honor and recognition in case of success. In case. And yikes, all right, that boat's not doing well. All right, so it's always been hard to get there. But when you do, there are fish that are only there. All right, I don't know that Shackleton saw them, but maybe. These fish that occur in the Southern Ocean, the first shows you that the notothenioid fish, which I'll talk about in a bit, are the majority of the diversity of fishes in the Southern Ocean. So it's like a handful of families that represent most fish that are there. The abundance, they are the most fish that are there. They eat krill, which is a lot of krill. Okay, that works. In terms of biomass, they are the majority of the biomass of fishes that are there. So why not study things that are that popular? All right, so the notothenioids. There are about 100 species of notothenioids in the Southern Ocean and some of the very southern temperate oceans. You can find them in parts of New Zealand and such, but then further south. They're related to perches, darters, and sea basses. That is, they're percomorphs. Okay, the Chilean sea bass is one of these. It is not a sea bass. Okay, it's a notothenioid. They are very adapted to cold water. They have a lot of adaptations for this. For example, they don't freeze. That seems good. All right, bluegill do. All right, but there's a little notothenioid hanging out in an ice sheet that if you touched it, it would freeze your finger. All right, the water is below the freezing point. It's salt water, and it's very cold. But they do that by having a lot of antifreeze glycoproteins that help. They basically can move water from inside their cell into the interstitial spaces and thus not freeze. Other cool things, there's one lineage that has lost the ability to carry iron on its hemoglobin, and it seems to have lost that before going into the Southern Ocean, so it's a pre-adaptation to cold water. They store oxygen in the plasma, not on the hemoglobin molecule so much, and their blood is white. That's the color of the gill. All right, whole lot of research being done on how that happened and how few mutations you needed to do that. All right, other fun things. Fish have swim bladders. If you caught a bluegill, there's a little gas bubble inside it to help keep it buoyant so the bass doesn't sink, or the bluegill doesn't sink. When the bass eats it. So that doesn't work in really, really cold water because you take a balloon outside and it shrivels up, right, in the cold winter. So you can't have a bag of air inside your body in sub-freezing temperatures. So they have adapted a few things like reduced skulls. Their bones are very, very thin, which makes them a little bit lighter, and a lot of fat in their muscle cells or between the muscles. And if you notice, these are histology slides, all right? And you may have seen that name Eastman show up a few times so far. Joe Eastman. He has had a career of over 53 years. He's still doing it. He has over 132 publications. He retired, I don't know, maybe 10 years ago, and since then multiple publications because, he has time now. He literally wrote the book on Antarctic fishes. The QR code will take you to a lecture on Antarctic fishes that Joe's giving. All right, so that's kind of cool if you want to see Joe. And he was on my graduate committee. All right. These are the sites Joe collected over his career. Or people sent him things from there. I don't know that he went to every single one of them. And it pretty much, I tried to do a GBIF output to overlay everyone else's collections, and it didn't work. But if you looked at it, you could see that they're more or less in the same places. So he basically has a collection that represents the diversity of places that people have collected fish from Antarctica. And I think this one is not on land. I think it's an inlet. Okay, unless he got it from a penguin or something. All right. In 2014, Joe donated his life's work. It's kind of like what happened. There are patterns, which is why we do these things. All right. And also how we do these things. So he donated a collection of Antarctic fishes and related material from Joseph T. Eastman. The collection includes archives and biological materials such as, we say such as because there were other things we didn't know about, dried skeletal material representing 24 species, 6 dried caudal skeletons, just the tail, of Dysosticus mossini mounted on cardboard, 94 cleared and stained specimens representing 38 species, approximately 1,200 fluid preserved fish, several hundred radiographs, 3 tissue block storage units, which are the embedded tissue you use to make the little salami slices that go on the slide. All right, or is it sashimi? So approximately 1,400 of those blocks are in there, about 150 microscope slides, each holding 100 slides, and the notebooks and the data, and he's still sending pictures once in a while. So the donation, we drove from Yale to there, and you did this slide too, but I don't have any animation. We got about that far, and the truck started bouncing up and down. So we stopped, and that's Tom Neer checking the tire pressure. It was not the tire pressure. We went back to the dealership. They said, "Oh my God, you could have died." And they held the truck for like a month, fixed it, and then we drove back, and we didn't die. All right, we scanned his notebooks in 2016. His notebooks included great drawings, latitude, longitude, who was on the ship, all kinds of detail. And we scanned them using, I think we used the Kurtas machine then, and it made a concatenated PDF of each notebook. And I sent Joe a copy of every one of those because he still uses them for his papers. I had a small grant to have every X-ray film or radiograph scanned by a medical X-ray company. When electronic medical records started, a lot of those old files had to be scanned so they could be stored somewhere. And it is very efficient. You get some pretty high-res TIFFs out of it. And literally, it was a week to do them all from the time I shipped them and got them back with the thumb drive. All right? And they charge per image. They don't care what's on it. And that was even with me telling them, you got to label the file names this way. So I slowed them down. And then we had a student, that one, spend several years of his undergrad career and the gap between undergrad and, I guess he had a job and then he went on to grad school. He went through all the records. He connected the dots between the X-ray, the slides, the fluid specimen, the cleared and stained head, the notebook. He literally read every single paper Joe ever wrote and all of his notebooks. And he figured out which specimens were which slides, okay, and which paper referred to them, which was no small task. But he started his career at Yale as a history major and became an English major. For him, he did it like in a weekend. But literally things like if it was Dysosticus from the Ross Sea, 1995. We may have only had 5 of those in the collection you gave us. It must have been those. And sometimes there were other details. Joe, any image that he used in a paper, he circled that particular salami slice on the slide, and he marked the edge of it with black marker. So the slide stands out in the box. And you can literally take that piece of slide and look at the picture in the publication and go, that's the same salami slice. All right, so that was the process. 2016, we did the notebooks first, so we had some sort of map to follow and figure out what's going on. 2017, we did the X-rays. 2019, we did all the whole chunks of fish and whole fish, and all that left was microscope slides and histology blocks, which were stored that way. All right, so we spent the summer, connecting all the dots, using Oliver's help. And then we got an IMLS grant, so that's a Yale, that's a U.S. grant for museums and libraries. And this gives us funding to do cleaning and fixing broken stuff, That's what Mariana will talk about. 2D scanning and rehousing of each slide into a brand new house, and updating the emu record to show where that lives now. So we actually did Oliver made a list of every single slide on every single specimen record. And then we wrote a script, well, actually Larry Gall wrote the script that went through, saw that there were histology slides, and busted out each one of those automatically into a child record for that specimen. So each slide has its own emu record, and carried over with it the number that Oliver said was written on the slide. And Oliver, he read them. He knew. All right. So right now we have done 14 notebooks scanned, 152 X-ray radiographs are scanned. That does not include weird things like bowling ball. He tested X-rays. There are weird things, a bouquet of roses for some reason, and a skull, a human skull. So that's just the fish. 249 specimen lots of 1,653 individual fish. That includes one that was a bucket of just heads of all the same species. I think they ate them. I do. Yeah. 12,259 microscope slides, total catalog records, 12,674 so far. And isn't that pretty? I think that's pretty. All right. Thank you.
Okay, we have time for questions. Any questions about the Fellowship of the Ring part of the program?
Can you talk about the resolution at which the slides were scanned? And if it's a large resolution, how are you dealing with storage challenges?
We don't worry about storage. Piper might talk about resolution and how big the files are, especially Z-stacked images that go to the virtual microscopy site, that sort of thing. Okay, but yeah, she'll stay tuned. Oh,
Did Dr. Eastman donate any samples, i.e. types, to other institutions in the past? If so, how does that affect your work?
did Eastman donate anything to other places other than just us? I don't think so. I think things from the cruises went multiple places. And it might be some of the histology slides I think we don't have a voucher, but that might be because he sliced up the entire fish. All right, so maybe. But once we get it all in the database, then we'll know, and maybe the other collection we can actually match and figure it out. But I don't think he did. He's been sending us stuff for years, and then all at once. No more? Should I walk off the stage slowly? Okay. Oh,
Wait, no, we still have.
Okay, don't do it yet.
I won't do anything.
Don't make her introduce herself.
We have 2 minutes, stretch, you know. Have I told you about our diamond sponsors? We must thank them. Please don't forget to visit the sponsor area and ask them all of your questions and
Get stickers.
yeah, all of the fun stuff. So yeah, we have 1 minute and 1 awkward minute of me standing here, smiling. No. Thank you also to our Zoom people for asking wonderful questions. We are happy that you are there. Okay, it is time for me to introduce myself. My name is Mariana Di Giacomo. I am the conservator at the Yale Peabody Museum. And I will be talking about the Two Towers portion of the program. I will be talking about the conservation aspect of the project, which is the second part of what we did in this project, in this grant. So just to give you a very quick recap, we got all of these slides donated with other things. There's a lot of data that happened, as Greg mentioned, and the problem were the slides. So before we got the grant, we were like, this is bad. We have poor housing. There's a bunch of boxes on top of cabinets. That is not good. We don't know exactly how many slides we have. Yes, Oliver did a lot of work, but it's one person doing a lot of work, and a lot of the times we all know how that goes. One person can make mistakes, and that is just normal, what happens. And so we had a very good number, but we had to go over that number. We had no clue about the preservation issues because Oliver is not someone who was experienced in that aspect of things. And the curation was not 100% proper because, again, boxes on top of cabinets, that's not good. And the boxes inside the boxes were like some horizontal, some vertical, scrambled. So we decided that we needed a grant. And for us to do that, we decided to do a condition report to justify this grant, to say, hey, we need money. Why do you need money? Bad. So we grabbed some random boxes from on top of the cabinets and opened a bunch of slide boxes and realized that, yes, poor housing and unknown preservation issues. I have had the opportunity to look at a lot of slides in my life, and I see Paramount from miles away, and I hate Paramount. And I was not very happy when I saw that, and I said, Greg, grant. So that's when we decided to really write this grant and get ready to work. So our goals for this grant were to get cabinets, clean the slides and fix any problems that we could fix, enter all of this information properly into the database what we needed to fix, and digitize the slides. And I have put the clean the slides and fix any problems in yellow because that's the portion of the program that I will be speaking about. So the workflow, what did we do? We got the slide boxes inside the cardboard boxes and cleaned the slides in order as we received them. We put the clean slides in trays and rehoused them in cubes. I will show you all of that later. We enter the information in an Excel spreadsheet for Greg and Piper, who will be talking about this later. We identified problems and fixed whatever we could, and we got more boxes and more cubes, and we kept the workflow going. And I say we, because we were extremely fortunate that in this grant, we were able to include money for students. And so all this work was done by students that were getting paid. We like to pay people. So that is the workflow. We received the boxes from Piper. Piper was here, boxes. And so this is a box that we have been already cleaning. But in the box, if the box was completely full, we just received the box. If the box was not full, sometimes inside the box, we got a little gift. If not, the gift was outside the box, which was that teeny tiny little bag that had a gazillion little labels. Those labels were supposed to go in our trays, and I will show you that in a second. So what we did was we grabbed our beautiful Kim wipes, we put on our gloves, especially because EHS was very adamant, environmental health and safety, that we needed to wear gloves to use our one-to-one solution of deionized water and ethanol, which means it's like 50%, which is less than hand sanitizer, which we were putting on our hands all the time during COVID, but it's fine. The amount of ethanol that we were using was more than hand sanitizer on a regular day, so safety first, people. And also, we did not want to leave fingerprints after we cleaned these slides, so gloves. So we're putting on gloves. We were using one Kim wipe that we were making wet we were cleaning the slide very well, and then we grabbed a dry chem wipe to dry the slide, and then you would use the light in the room to remove all the smudges and leave it absolutely perfect. And don't put your thumb in the pencil, because bad. You will erase all of that very important writing. Then we would grab Piper's little labels. She would print all these beautiful labels for us, and then we would put them on the trays. And then when we did that, we would put the slide, the very, very clean slide, on the tray. Look how pretty all of that looks. We would also label each tray. We printed a gazillion little labels, and we labeled each one of these trays, and then we put the trays in the cubes. And then these cubes, we would fill the cubes, and then we would give the cubes to Piper. And also, we had a couple casualties. Actually, couple, because only two. Yes. I show you one of them. It fell, it broke. So that is one of them that we kept, because we will see if there's anything we can do about it. I doubt it, because it's pretty bad. But you never know. And we also kept the bags. Every teeny tiny little baggie that Piper gave us, it's in that bag of baggies. Why did we do that? Because I'm not throwing anything out until we're 100% done with this. Why? Because we have had to go back to the baggies, because sometimes there were some labels left, and so there were some issues with some of the data and stuff like that, and we have had to go back to the little baggies. And so we have not thrown anything out. Sometimes being a hoarder is a good thing. We also left a lot of messages for Piper in my whiteboard in the lab. Sometimes we didn't have labels because, again, the data wasn't perfect. And so the labels were printed, the little paper labels were printed, and we actually had more slides than labels, or we had more labels than slides. And we had the broken one, and we had some, sad faces. The students would love to do the sad faces when there were no labels. And we had the Odo, that is Oliver D. Orr. He labeled each box Odo 1, Odo 2, Odo 3. So the boxes are the Odos. And so we were able to talk about the trays and correlate them to the Odos and all of that. And we have a spreadsheet that correlates the specimen to a lot of information. And we're able to also say if there's a chip on the slide already, if we have any issues with the slide itself. So we have this little spreadsheet. Then when we're done, we're going to tie it to the EMU record as well, the database record. And we did find some problems, because we knew that we were going to find these things, because when we did the initial condition report, we found this stuff. The problems were essentially two. Yes, we found some chips. Glass breaks. That's what glass does. It's very delicate. It does that. The problem is not necessarily that, because a lot of the chips happen on the edges of the slides, and they're on the data side, and it's usually very small chip. And so it doesn't remove the data. It doesn't create usually dissociation or any problems like that, because that's the weakest part of the slide, because it doesn't have the sandwich. So usually that's the case. It's not a big deal. However, we had some issues with the mounting media. These are the two problematic ones that we found. So I'm going to start with my nemesis, the yellow one. When I identified it at the beginning, I looked at it and I'm like, that's per mount. That's per mount. And again, once you've seen this, you can identify per mount. Anybody who's been working on slides for a while, you see this milky white closing in on your slides and just erasing all of the data, and you see per mount. So I took a sample from, I'm going to go back for a second, from the outside of the slide. I know I have a thing, I'm sorry. That, of the slide, and I took it to to be analyzed. And it was just, identified as some sort of natural resin. And I knew it was Paramount. And then we had the confirmation from Joe Eastman. Oh yeah, I use Paramount. And then we had, I'm gonna go back again, this one, which to me is 10 times worse, because the cover slip, the histological cuts, and the mounting medium are detaching from the slide with the data. So you have a huge dissociation issue, and it's happening on the whole series, all 100 slides from that box. So, and it was happening, in all of them. And so what we found is that it is this cellulose nitrate with some weird organophosphate situation going on in there. So what are we doing about that? For Paramount, there's nothing else but scanning, because that beautiful mounting medium is only soluble in toluene, which is terrible. And it is extremely hard to unmount a histologic slide. If it was a full specimen, something like that is a little easier. But histological cuts, it's very, very hard. But for the cellulose nitrate, I'm testing some chambers, some ethanol chambers and stuff, and I'm able to just glue it back on. But yes, I'm still doing some of the tests to see if we can actually bring those things back on and not have to worry about the levels of dissociation that we're dealing with. And as I told you, I give the cues to Piper, and after any questions that we may have in the minute that we have for questions, I will give Piper the Return of the King part of the program. Thank you very much to the conference organizers, my colleagues of Yale IPCH for the analyses, my colleagues, of course, at Yale, and all of you for listening. Time for one question. There's one online?
Yes. How many different mounting mediums were used in preparing the slides, and does that affect future curation?
How many mounting media were used in the preparation of the slides? The problematic ones, so far we saw 2. I don't know if there's more than 2. There are a lot of slides that don't have problems. That doesn't mean that they are either of these 2 or a third one or a fourth or a fifth that don't have problems. I don't know. We still have to test. I started testing the problematic ones. I would like to test the non-problematic ones after. So. All right, we have 2 more minutes. Any other questions? Okay. Yes. Microphone, please, because our online audience otherwise cannot hear.
Hi, wonderful talk. Just a very brief question about if you did have to dismount and disassociate those, what is the approximate time it would take for the perform slides to have to be disassociated and rebuilt again?
So the ones that are already dissociated, I don't have to do that. It's already done for me. But the chamber, I put the slide in that chamber for a week and a half, and it's still not tacky enough for me to be able to put it back. So I'm going to be doing a lot of testing so I can develop a technique that allows me for this to happen faster. I didn't want to put anything directly on the mounting medium. I wanted it to be an environmental thing, because I don't want to do anything to the stain. I don't want to do anything funky to the cut. And so I just wanted it to be in the environment. I'm going to be doing a few more tests, but yeah, it was there for a week and a half, and it gave me a little bit of tack. So that's going to be a fun next step. All right, we don't have any more time. Thank you so much, Gary, for the question. Now I'm going to be inviting Piper Stepule to the podium.
Hi, this is the 3rd presentation on this grant. I did the digitizing part of this project, so I have, at this point, scanned digitally about a third of the 12,000 slides, and it's been a year. And we have a ways to go. So topics for this, so we're gonna talk about why we digitize, as if you don't already know. The machine itself, the slides themselves, and the problems that I've run into and their solutions. First of all, I'm Piper. I work at the Yale Peabody Museum. I'm the museum assistant for the vertebrate zoology collection. I've worked in this collection for about 4 years now, and like I said, I've been working on this project for about a year. So first, we're going to talk about why we take the time to digitize these slides, starting with it makes it internationally available data for everybody without risking damaging the slides by sending them across the world. It optimizes specimen loans by maybe even making them obsolete. They may not be needed at all if we have a proper scan of this slide and if that's good enough for research. And it's a way to permanently preserve the data in case of preservation failure. So the machine and the slides themselves. So this information was courtesy of Eric Lazawazam of the Invertebrate Zoology Department. He is the one who takes care of the machine. It is a very fickle machine. It breaks very often. We don't really understand why. But it is, so the microscope itself is an Olympus BH51 compound microscope featuring lenses that allow both transmitted and reflected light. It has a Infinity 3 digital camera with positioning of the slides that focus and the image acquisition were controlled by Objective Imaging Surveyor, the software, along X, Y, and Z axes. And the individual position images were stitched together on a single plane if they were using a Z stack. So taking a scan at several different Z layers. And they are squashed together using a software called Helicon Focus Pro. And then the final images were processed in Photoshop. So we're going to talk about the difference between a quick scan and a Z-stack scan. So one that's just you pick one Z level and you scan it that way, or you pick several Z levels and scan several layers and squash them together. So on a very quick look, it doesn't look like much of a difference. So this slide, for example, the specimen part is about maybe 2 inches by 1 inch. So it's a very big slice. But the problem I was running into was that they weren't mounted flat. So they are very flat slides. So if they are mounted flat, I don't need to do a Z-stack, but some of them are just a little tilted. So if this is good enough and you just want to see what the slide has to offer, this will be great if for a quick scan. However, when you get closer, you can see that this side is very blurry with the quick scan and that side is very clear. But in other parts of the slide, they will look about the same. So quick scans are great if it's a very flat surface. Otherwise, a Z-stack is pretty important. But even so, like you can still see some distinct blurring on this first slide. So the slides themselves, as Greg was saying, there are over 12,000 slides. They are stained in various different ways and with various different preservation methods and states of preservation failure. So generally like I said, generally the same thickness. They're very, very thin, but they don't sit flat, all of them. A lot of them do, thank goodness. But yeah, and then as you can see from this, there's indicators of specimens of importance. So let's talk about the bumps. So the problems I was having with, first of all, the machine itself, you do a lot of colorations, shading adjustments. So between every different color would have to take several minutes to adjust the coloration, adjust the shading. The size was also causing lots of issues. As you can see, there are, let's see how you do this. So very, very, very tiny little dots on this slide and all the way to like very big slides. So, something I forgot to mention. The big difference between a quick scan and a Z-stack scan is the amount of time it takes. Quick scan, about 30 seconds, if for a specimen that big. A Z-stack, probably about 20 minutes. So, and I only use about 5 steps, so 5 Z levels, and it takes way longer. So if you can get away with a quick scan, it is very time efficient. But so the size variation, the color variation, and made it so that you had to take time in between specimens to make sure that the image was good and usable. And then also, as Mariana was touching on, the levels of preservation failure, I had to get a little creative when it came to scanning. So this is the lifted slide covers. And the way I was doing this was double-sided tape. You take a blank glass slide. I was flipping over the popped off cover slip, double-sided taping around the edges of it to make sure nothing was touching the specimen itself, and I'd scan it upside down to get kind of the same result, practically. It's technically upside down, but the message was received. And the amount of these is so annoying. There are so many that are just like this, and they eat up time. And also because of the strange way I'd have to mount it, it made Z-stacks more likely to be needed. This is a slide of what it looks like when there was no mounting medium and there was just specimen on glass. It doesn't look great. Scanned it the best I could. It looks like shattered glass. There's not much you could do about this. So you just scan it, accept it the way it is. This is an example of a currently failing per mount slide. So as you can see, if it's fully failed, it looks like that, which is unusable. But for a lot of them that were halfway to failure, I was able to scan what I could, get what I could out of it, make a note of it, move on. As of right now, about 1% of the entire lot is per mount and completely unusable and looks like that. So that's 1% of the total lot of what I have now. So it's probably going to increase, but it'll be around probably about 3% by the end of the project, which is not terrible, but not great. And that's all I have. I went very fast. Is there anything online?
Please go ahead, ask a question.
Yes,
I'm going to be
please.
sitting there.
Yeah.
It's the same question I had for
Yes,
Greg, but-
as soon as I stopped talking, I remembered that you had asked that. So, yeah, that was another huge problem I had run into. So I forgot to mention this in the presentation, but so I used about a 10x magnifier, and then the raw images after squashing them down with the Z-stacks came out to be like 1 to 2 gigabytes, which is not. No, it can't happen. We were having a lot of issues with the database getting clogged up with uploading and like just crashing a lot of things, and it just wasn't going well. So Larry Gall, Lawrence Gall, wrote me a script that it takes all the images, it runs them through a LZ something, and yeah, and it greatly diminishes. Huh?
Can you repeat that for the people online?
It takes the TIFF file, makes it LZW compression, and compresses it to about like 1,000 megabytes maybe, or like under 1,000. So and it makes that I was having the problem where I was limited to 100 slides uploaded a day because it was just like messing up the ingester and it's just not going well. But now I can do as many as I want because I have Larry who just squashed them all down for me, and it also makes my life so much easier because I was doing that manually before, and apparently I wasn't compressing them enough. So, yeah.
How do you determine the number of photos necessary for a Z-stack?
How do you determine the photos necessary for a Z-stack? I was just going general 4 to 5 step rule because they are very thin, and I was consulting with Peter Griffin? Oh no, Chris Griffin. His name Chris Griffin. He was the one who helped me with the slide scanning he's a PhD student. He taught me how to use the slide scanner, taught me how to use Z-stacks, and he said a general rule is about 4 to 5. And if anything more than that, it would just kind of be unnecessary. And also for I just did one piece per scan for slide.
Hey, it's a very general question, I guess. And hard to answer.
Okay.
I guess I'm wondering, you were talking about the color variation, and I'm wondering if there was any advantage to like pre-sorting and having all the similar colors together, and if that was even doable.
Yeah, that is doable if we had like maybe another year. Because right now we're really just kind of shoving them in the boxes as they come along. There would be advantage to that for sure. But it'd be very hard to keep track of.
Yeah.
Yeah.
Can you talk a little bit more about like the database end of things? So you decided, if I'm correct, to do one slide per record in KEMU, or did you do multiple slides?
It's just one. So each specimen has a parent record, and then there's however many slides. If there's like 100 slides for that specimen, they each get their own personal record. So each microscope slide has their own record of one chosen specimen of that slide.
And then you can relate those together in the
Yes,
series.
and they're all nested in together, so you can go through the whole fish.
How long does it take to make a scan? Max average.
How long does it take to make a scan? So if it's tiny, like those little dots, literally 1 second. So if it would be really small, just doop, and then move to the next one. So like 1 second, maybe 2 if you count me changing the slides. The bigger the longest a Z-stack has ever taken me was 30 minutes. So anywhere from 1 second to 30 minutes. I'd say the average is probably about 30 seconds.
Apologies for a repeat question, but do you guys know who's using your images? And what for?
Greg, do you have an answer for that? Microphone.
So currently the images, I think the next day they appear on the web page for the museum. So it's an overnight upload, and I know we can actually do some sort of Google Analytics and figure out who's doing what, or at least who's going there and where they're coming from, but they don't have to ask to download it. They don't have to ask to go to it.
Right.
So unlike,
Universally available.
yeah, so unlike like CD scans where you have to actually ask to download, these are free. So they're using them for everything. I don't know.
Anything else? Anything online? Great. Yay, thank you.
All right, now people can stretch again. Relax for the next 2 minutes. I'm not going to introduce you yet. Yes, stretch. Dance. And remember, we have this 15 minutes at the end to, generate discussion. We can talk about any topics related to microscope slides that you've been really wanting to talk about. So we will have that opportunity at the end. And of course, don't forget to visit our sponsors. Oh, and I had a question online. I'll just answer it. I use FTIR, Fourier Transform Infrared Spectroscopy, for testing the mounting media on my slides. Waiting patiently. Yes, Bill. I tried at Smithsonian and it didn't work. They were big, so maybe for smaller ones, yeah. Yeah.
Cut away the fur mount, and then you put like something like Canada balsam, but Palomar balsam around the fur mount, and you put
Yeah. But for histological, I haven't tried because I'm terrified.
You're gonna have some
Left behind.
left behind, some on the cover slip, so that's not really good.
Fear. Okay. Please welcome Elise Chimat to the podium.
Thank you. So I will be talking about mounting medium degradation. I'm working at the Center of Research for Conservation in Paris. So today I will focus mainly on two main mounting medium. The first one is natural resins such as Canada balsam. So chemically it's mainly composed of a mixture of terpenoids compound, and the second one will be about synthetic resins such as polymers obtained by man-made synthesis. So if we look now to slides and damage that can occur, you are all familiar with this kind of aspects of the slide, I suppose. So you can have several types of damage on the slide, such as yellowing of the mounting media or darkening of the mounting media, especially in the case of gum chloral mounting medium. But also you can have some cracks or a drying of the mounting medium or even liquefaction of it. So if you want to know more about the aging of the mounting medium or about mounting medium chemistry, you can have a look at these references, and the later one will be published maybe next year, I suppose. It's currently under writing with the colleague of Germany. There are several factors that can affect the aging of the mounting media. And I was mainly wondering which mounting media we could choose to mount in the future, and maybe even to remount specifically gum chloral mounting medium that have been damaged. So I suppose each one of you have your favorite mounting medium, and it's a bit like this in every field. But I wanted to compare under controlled conditions how they behaved under aging. So I asked entomologists what they lacked, and I decided to choose 3 mounting media to be studied in this current study. So the first one is, of course, Canada balsam, which is natural resins, and it's a bit of a reference since we have used it since at least 100 years. And so it's composed of this molecule, basically, so terpenoid structure. The second one I chose was apparel, which is a mixture of natural resin, sandarac resin, but also with other compounds such as camphor or eucalyptol, for example. And the last one was an acrylic mounting medium, so named Urkit, but you have also other brand names. So in order to compare those mounting medium, I had to prepare mock-ups samples, so without any specimen in it first. So I decided to use a coating applicator, and so in order to have most homogeneous sample I could get, and I let them dry on a hot plate before conducting my artificial aging. So now what about the artificial aging I chose? So first I used a thermal aging at 70 degrees, and secondly I used a light aging, so in order to see what were the parameters which could affect the damage of the slide. And to monitor the sample, so I did do some colorimetric measurements, but also carried out some chemical analysis. So for example, infrared spectroscopy, mainly to have major composition of the sample, but also some chromatographic analysis to have access to the molecular level of composition. So what are the results? So yes, I conducted the aging during almost 50 days, and it was kind of really accelerated aging. So you have in the middle the reference sample, so sample that have not been aged. And on the left-hand side, you have the thermal aging results. And you can see that kit is still pretty transparent. And that apparel and Canada balsam are yellowing. And Canada balsam seems to be yellowing a lot, but it's also a bit thicker than apparel. And if you look on the right-hand side, you have the results of light aging. So you can see that there is also a slight yellowing of apparel and Canada balsam if you compare it to the region where a mask was on. So now, what about the colorimetric measurements? So to have more quantitative data, you can see that clearly with the thermal aging in red, there is an increase in the yellowing of the sample, both for Canada balsam and apparel. Once it has been normalized to thickness, it's similar. And for the light aging, there is only a small yellowing for the apparel at 50 days. Now, what about the cracks formation? So for Canada balsam, we did observe a lot of cracks, both for thermal aging, but also for light aging, even with bubbles formation. And for apparel, it was only for thermal aging, the cracks appearance, not for light aging. Now, in the following slide, I will focus mainly on Canada balsam, and you can have a look a bit more to chemical changes, so don't be afraid. So those are infrared spectra. Basically, what you need to compare is a black line, it's a reference line, with the red line, it's after the thermal aging. And what we can see is the increase of these two bands here, mainly, and some shifts in another part of the spectra. And that means basically we have formation of oxidized and aromatic structure. With light aging, mainly the same trend, but with much more change in the spectra. Now, if we look at the chromatographic analysis, so I told you on the first slide that we had this molecule, the abietic acid, at the top of the slide, in the Canada balsam mounting medium. And when there is oxidation reaction, it will transform step by step until it gets at the bottom of the slide. And so the further we are, the higher the degree of oxidation we will get. So based on the result I had with the chromatographic analysis, I get the degree of oxidation level to have a quantitative number. And as for the colorimetric results, there is a higher degree of oxidation for the thermal aging than for the light aging, which is almost similar as a reference. Now, this number, 0.3, what does it mean compared to real Canada balsam sample that have been naturally aged? So I did the look on 3 mounting medium I had naturally aged in several labs, but with unknown conditions. And so it's pretty similar value around 0.3, whatever the age. But we don't know in which condition they were kept. So. So it's still a study under progress, but what I can say so far is that for a kit, there is almost no change so far. And that, of course, for natural-based resins, there are much more changes, mainly due to temperature. So if we think of using those mounting media, we also need to think of what about their future solubility or maybe melting ability if we need to remount, for example, the slide. So regarding the melting temperature, it's quite high, so I'm not sure we could use heat to restore some slide because the specimen should be able to go at 60 degrees. And the solubility, it's something I have to test, but we can already think of the presence of some polymers in apparel and a kit that might prevent the medium from solubilization. And of course, you have to think also about the practical aspects you are looking for. The two first medium, Canada Balsam and apparel, are slow drying. Canada Balsam needs a lot of dehydration and cannot be used with cationic dyes. So yeah, and a kit, for example, it's fast drying, you can adjust viscosity, but there is sealant, which is toxic inside. So you need to balance between the durability and what is really needed. But so the take-home message I have is everything aged. It doesn't mean necessarily that the specimen will be in danger. So here I have some example of specimen found in the micropaleontological collection that are still pretty good and well preserved. And yeah, please think about the mounting media and its permanency before starting mounting the slide and try to document everything. Thank you for your attention.
Thank you, Elise. Do we have any questions? I do. I have so many questions, but we'll talk later. But we're going to nerd out together later. But I have one question that I find very interesting. So have you thought about, because what you were saying about the cracking, that is something that we see, you know, when people squish the mounting media and they leave all the extra mounting media outside the cover slip, we see the cracking. Have you thought about doing all the aging, but with a cover slip?
So I did not try with the cover slip because it was not very convenient for the analysis I had to carry out after. So we could do it, but yeah, I'm not really sure if it's coming from a kind of pressure or stuff like this. What I think is that we could have some plastifier or molecule acting as a plastifier that are evaporating, and so we have cracks like for permount. But I didn't do the analysis yet, so but I could also try with a glass cover slip on.
What influence might fluctuating humidity have?
What influence might humidity have? Something like that. I'm not so sure because for glass slide collection in general, you have cover slip, so it might not be as affected as other mounting medium. It also depends on the types of mounting medium, and especially for glycerin jelly mounting media, I think it will have a kind of impact, but it's also always going pair with temperature changing. So, but yeah, it could also mainly influence the liquefaction of some mounting medium, relative humidity. So.
All right, thank you, Elise. Okay, our last talk of the session is by Miriam Hebert, who's actually online to answer any questions that you may have, but we will have her presentation on a video right now. So let's hear from her.
Miriam Hebert, and I am a contract researcher at the Smithsonian Institution. I'm sorry that I was not able to be there with you in person this year, but I'm grateful to have the opportunity to talk to you about the work that my colleagues and I at the Smithsonian have been working on over the last several years. For those of you who may not know, the Smithsonian Institution is the world's largest museum, education, and research complex. We have 21 museums, 9 research facilities, and the National Zoo. And we're located primarily in Washington, D.C., with satellite facilities in New York City, Boston, and Panama. We house the National Collection of the United States, which is comprised of millions of items spanning all of human history and knowledge. This includes the National Museum of Natural History, which is the collection that I will primarily be talking about today. I'm a member of the Glass Deterioration Working Group. The GDWG was formed at the Smithsonian in early 2020 and took shape largely over the course of the COVID pandemic. It's a large-scale, pan-institutional, collections-focused statistical survey of the current condition of glass collection items across the Smithsonian. As far as we are aware, this is the largest scale survey that has ever been conducted in any cultural heritage institution, and developing this project has taken a great deal of work from a dedicated group of people, including curators, conservators, and collections managers at the Smithsonian and from external cultural heritage institutions as well. The breadth and depth of the Smithsonian's collections makes it the perfect laboratory for understanding glass on a broad scale. The first phase of our project was focused largely on glass collection items in each of the Smithsonian's collecting units. This included types of glass objects that may be the first to come to mind in a discussion of glass in museums, so archaeological glass, historical glass objects, beaded objects, contemporary sculpture, etc. However, as you are all very aware, glass is not only found in museums as part of collection objects, but also plays an integral role in the storage and preservation of museum collections, particularly in natural history collections. This includes fluid specimen storage, as well as microscope slides, which have become the focus of the second phase of our work. One of the most surprising things that we quickly learned about storage glass is just how much of it there is. You can see in the pie chart on the left that even only rough estimates of the number of glass storage containers and microscope slides that we have available to us indicate that this type of glass vastly outnumbers the glass collection items that are found across the institution. What's even more striking is that the majority of this storage glass is found in the collection of only one of the Smithsonian museums, the National Museum of Natural History. In the pie chart on the right, you can see how the approximately 1.3 million microscope slides that are held at Natural History are distributed amongst the different departments. The two largest collections of microscope slides are found in paleobiology and invertebrate zoology. For the purposes of this study, we will be using invertebrate zoology as a proxy for understanding microscope slides across the entire museum. This is due in part to the generosity of the team in invertebrate zoology, who have given us access to their time, expertise, and collection, but also because the slides in invertebrate zoology span a wide range of different time periods and preparation techniques, making them representative of slides across the institution. In our conversations this morning and in previous studies and publications focusing on the condition of microscope slides, there are a number of conservation concerns that are typically discussed. Chief among these is mounting media deterioration. We are very lucky at the Smithsonian because the condition of mounting media on slides at the Smithsonian has been particularly well documented as the subject of an immense study that Mariana undertook a few years ago. Other concerns that are typically discussed are breaks or cracks in the slide itself or in cover slips, and also label or sample loss from the slide. What has not been as much of a focus is the deterioration of the glass itself. So for those of you who may not be familiar, glass deterioration, also called glass alteration or sometimes glass disease, is a form of chemical deterioration that occurs in glass over time when water or other hydrogen-bearing species in the surrounding atmosphere undergo an ion exchange reaction with the alkali ions that are present in most glass matrices. This leads to the formation of secondary precipitates on the surface of the glass, as those alkali ions are drawn to the surface of the glass and crystallize, as well as in the formation of a depleted altered layer on the surface of the glass. This layer takes the form of a hydrated silicon oxygen gel and leads to the characteristic crizzling or haziness that is observed on glass bodies that are undergoing significant amounts of alteration. The rate of glass alteration that a given object experiences is largely dependent on two factors. One is the glass composition. Higher alkaline content in glass will lead to faster rates of observed alteration, and we know that glass slides are primarily comprised of two typical glass compositions, borosilicate glass and soda lime glass. The second is storage environment. Higher humidity environments or microclimates can lead to an increased observed rate of alteration. The images you see here are of collection objects that we've encountered which are experiencing various symptoms of glass alteration. Presentations of glass alteration in typical collection items have been very well documented. What hasn't been as well studied is how this phenomenon presents in microscope slides. Through our survey of glass microscope slides in invertebrate zoology, we've encountered a number of slides that show clear signs of this form of deterioration. Typically, what we've seen has been the secondary precipitates forming on the surface, either as crystalline deposits or in situations where a microclimate has been able to develop, we also see the formation of liquid droplets, or weeping as it's sometimes called. You can see images of how these two phenomena have appeared as we've encountered them in the collection. One aspect of developing a survey of the condition of glass in microscope slides that has been particularly challenging is accounting for the wide range of variability that we've encountered on these objects, from variations in size and preparation techniques to a wide range of materials that are used to mount different types of specimens. We've found that about 15% of the slides we've encountered have fallen outside of what might be considered a standard microscope preparation. As we've developed the survey for this collection, we had to keep all of the different aspects of these objects which may play a role in the observed glass deterioration in mind. The first step in this survey process was obtaining an accounting of the total number of microscope slides that are held in invertebrate zoology. This was obtained through a combination of collections database information and hand counting. In total, we've determined that there are 444,261 slides held by invertebrate zoology. Because it would be impossible to look at each of these slides and assess the condition of the glass, we are conducting a statistically randomized sample survey of this collection. We will be surveying a total of 767 of these slides that are held at 2 locations at the Smithsonian. Thus far, we've completed our survey development and finalized the reference manual for filling out the survey form. We've also completed the survey for all of the slides at one of these locations, totaling 395 slides surveyed so far. Our survey of microscope slides, as with our survey of collection objects more generally, has relied solely on visual inspection in order to determine the condition of the objects in question. In some cases, the presence of glass alteration is abundantly clear, but often whether or not a piece of glass is actively altering or is perhaps just dirty or scratched can be a difficult determination to make. Collection storage and care can directly impact our ability to identify altering glass, as older storage containers often lead to more dirt and dust on a slide surface. It's also worth noting that cleaning practices and object use is different for slides than for more typical collection items. If a slide is being regularly pulled for use by a researcher, they are likely to be cleaning its surface on a regular basis, for example. These factors make relying on visual inspection tricky. However, each slide was assessed before and after cleaning with a Kim wipe, which allowed us a greater degree of certainty in our results. Interestingly, we found that half of all microscope slides that we surveyed showed some degree of glass alteration. It's also worth noting, however, that this deterioration seems to be largely contained within specific parts of the overall collection. So, for example, there's a large number of cephalopod histology slides, which make up almost a third of this sample set, many of which were observed to have similar precipitate formations on their surface, potentially due to the glass composition of these specific slides or to the unique arrangement in which they are stored. Slides that were made and used at a similar time and in a similar way are more likely to be deteriorating in a similar way as well. Keeping in mind that these are only preliminary results, based on these numbers, we'd expect that about half, or about 218,000-ish slides in invertebrate zoology will have some form of visible glass alteration or deterioration. Just for some added context, I thought it would be interesting to note how these initial results compare to our initial results from the broader collection survey that we're conducting simultaneously of more traditional collection items that contain glass. We're a little over halfway done with our survey of glass collection items and have looked at a little over 3,000 of these objects. And we found that about 45%, plus or minus about 1.7%, of glass collection items have indications of visible glass alteration, which is slightly lower, but still within the statistical margins of error when compared to the prevalence of glass alteration we've observed in microscope slides. So they're altering at about the same rate as collection objects. Through the survey, we've also been tracking instances of other forms of glass deterioration that are important for slides, including the presence of breaks, chips, or cracks. Interestingly, we have found a much lower rate of this type of deterioration, with only about 4% of the slides that we have looked at showing any significant breaks or chips in either the slide or cover slips. And we've also used this opportunity to track instances of missing or loose labels on the slides that we've surveyed and have found a relatively low prevalence of this particular condition issue as well, with only about 3% of the slides we have looked at showing any signs of loose or missing data labels. This project is still ongoing, and there is still a significant amount of work remaining to be done. The next steps in our immediate future involve continuing and completing the survey of microscope slides in invertebrate zoology, and this will include compositional studies of a small number of these slides as well. We also plan to identify and survey any other groups of microscope slides that are relevant to include in a sample set that is representative of all microscope slides across the Smithsonian, so petrology sections, for example. And finally, we plan to compile our results and recommendations for slide storage and preservation both at the Smithsonian and in other collections that store microscope slides around the globe. This is a massive project, and we could not do it without so many members of our support team. We are grateful to the folks at the National Collections Program at the Smithsonian, particularly Bill Tompkins, Samantha Snell, and Amelia Kyle for their support of our project for the last several years. We're so grateful to all the folks at Invertebrate Zoology who have allowed us access to their collections and given us their time. And we couldn't have done any of this project without all of our GDWG members and external collaborators. We're grateful for everyone's effort in this project. If you would like to know more about this or have any questions, feel free to contact either Rebecca Kaskowski or myself at the emails listed here. And I really appreciate being part of this conversation this morning. Thanks so much.
All righty, thank you, Miriam. She's here in the Zoom. So if anybody has any questions online, we will still read them for the people that are in person. And if people in person have questions, please reach to the microphone. Piper, can you please tell us?
Can you provide figures for high humidity environment damaging glass slides?
Can you provide figures for high humidity environment damaging the slides?
Figures in what way? Like estimates of what humidity is detrimental? Is that the question?
Or maybe which humidity levels? Okay, yeah.
So that's a tricky question to answer because on some level, any humidity that is in contact with the glass surface will lead to some form of deterioration over time. So any humidity is too much humidity, but of course, when you're talking about complex objects that have other materials, including the mounting media, just for one, that's not a reasonable way to store these objects. So it's really up to conservators and collections managers to find the lowest humidity that works for the objects in question. So yeah, it's a really complicated question to answer for a lot of different types of glass objects with other materials that are involved.
Any other questions? We have one more minute. I have a question. Do you have any suggestions for solutions?
So one of the things that we're finding is that the way in which slides are stored seems to be important for when we're seeing the formation of the secondary precipitates and those types of issues. There's a massive project in invertebrate zoology to move all of their slides into the beautiful white new cabinets. So rehousing is a really great way to start. I know that that's a tall order for these collections where there's so many objects to organize. But even just having some separation and some airflow between the objects and not stacking them or shoving them together really makes a big difference. Also moving them out of maybe old boxes that are made of wood or plastic that's deteriorating in different ways that can be detrimental to the glass. That also is a helpful way to start handling this.
Thank you very much. Okay, so that concludes the talks portion of this session and begins the discussion portion of the session. And what I would like to do is invite anybody who may have any questions about any of the talks that we've had, or any questions that you may have about either your collections or anything regarding microscope slides. Please go to the microphone or ask your questions online, and let's start a discussion about how we can protect microscope slides for the future. So let's just start a conversation, and anybody can answer and anybody can ask. So this is just a discussion for all of us in the room. So one question that I have as Carol is walking is actually for you, Stevie. My question for you was, do you have a record of the mounting medium that was used in those slides that you received? And are you incorporating that in your database?
Yes. So as I said, we were fortunate that he provided us with like 7 notebooks, and some of them were the specimen preparation notebooks and some of them were the slide preparation notebooks. So he's like very detailed, said what is what. And that was part of the game of like, what is this thing? Because he didn't ever clearly say mounting medium. So you have a bunch of us who are just like, I'm a herpetologist. What the heck is this chemical? So we do have that information. I sadly don't have it off the top of my head right now, but we will be incorporating that into the specimen records. So that little attribute, I think your eyeballs popped up when I put that attribute part onto my slide too. That was just a screenshot from a different collection, but we do have the capability of putting that information, and we will be putting that information into the specimen records because of these conservation concerns.
Cool. Carol.
Okay, I came to this session because I have a problem, a big one. I've inherited 3 different collectors' slides. And some are, they've got various mounting mediums, they have lost or detached labels, and then the question is, you know, those little old labels that they used to lick and stick on there, how can I reattach? What kind of glue medium can I use to put those back on? I've got chips and broken ones, I have thin sections to things, objects that are practically 3D, because like Jose Cuatrecasas did some thin sectionings, but then you had Harold Robinson who looked at flower parts or leaf parts or whatever. So you got those type of objects. No notebooks. We are running blind with this one. The data's all on the slide. And in case of Vicky Funk, she etched onto the slide some number, which I haven't quite figured out what it is. I think she might have a notebook, but I'm not sure about that. I have various storages. I have boxes, I have trays, I have cardboard where they're you open up the 2 flaps and they're lined up. And so I have that. How can I capture the data? So I don't want to, eventually this collection will be removed from me, taken from me, and given to somebody else to image. But I want to make sure before it disappears that it's captured. What data is critical for me to capture? Now with Harold Robinson and even with Cuatrecasas, there could be types in there as well. So I need to know what data is important, how can I capture it? Excel spreadsheet, I'm happy with anything. I do emu, I'm not not a problem. And barcodes, because I want to be able to relate this one slide to this record in emu. I want to be able to somehow make sure that that data is captured and organized. So you can see where I'm at. Let's start from the beginning. And I do have cracks. I do have in the median, I do have it like Hoyers sometimes. Oh, I know I have Hoyers. So I don't know, you didn't talk about that one.
Yes,
Probably,
a little bit.
A little bit. Permount probably in there. I don't know what the other ones were. I don't know what Jose Cuatrecasas did, 50, 60 years ago. When you have the median is starting to shrink under the cover slip, they used to be able to, I know they used to take fingernail polish, there's probably that in there too, and put fingernail polish around the edge to keep things from slipping in, from drying out.
Doesn't work.
Is there a way
It doesn't
of?
work. Yeah.
I got all the fun stuff. Yeah. Is there a way of, and I know some have done that, is there a way of putting like the median along the edge and let it somehow suck its way in? Is that possible? So I need help. So just launch me, the first step is what, and how can I
Well, it depends
do
what you want to do first. But I think the first step is just like step back. Take a breather, Carol. And decide what your priorities are. And I think the data is definitely the first thing that you need to do, because you cannot start preserving something that you don't know what it is and what you have. And so the data portion of things, I think is the most important aspect. Just try to identify what you have, how many of what you have, how many storage containers, and the variety of your, because if you have the beautiful, I love those paper cardboard things. Love them. I know I'm a conservative, I'm not supposed to say that, but I love those. They are historic, they're beautiful, they just talk about so much about the history of natural history. Yes. So, but I understand, the importance of rehousing and all of that. And I think that is your first step, just taking a step back and assessing the situation. And then once you know what you have and what you need to do in that, aspect of things, you will start deciding how many broken things you actually have. Because sometimes you start opening things and you see a few broken things and you freak out and you get overwhelmed and you think you have more broken things than you actually do. So getting actual numbers is a very important thing. Just opening the boxes, doing an assessment, doing some statistics on the situation is a very important thing as well. And the other thing that I always like to tell people is perfect is the enemy of good. Do something. Something is better than doing nothing. And so just having an assessment and having that first step is better than nothing. If those labels that you are struggling with right now, because they're popping up and then you don't know what to do with them because you're afraid that,
I'm never afraid. I'm gun-ho.
no, afraid that you're going to lose them,
I don't want to lose them.
Exactly. You're going to lose them. You're going to lose the data and all of that.
Right.
Well, you have wonderful conservators at NMNH, and you can consult with them on either an adhesive or a non-adhesive method. Maybe there is a little sleeve, something that you can do to hold that label in place for now,
Right.
until you can tackle it in the future. Because it doesn't have to be tackled right now. You can put a stopper on the situation for now until you can address the whole situation later. Maybe it's with a grant, maybe it is with something else, but doing something right now to assess and take those first steps is what I always tell people, to start with something that also eases that stress and makes a ginormous project into something that is manageable and it's doable. And I always say the same thing. We have job security, people. Collection work never ends. Yeah. And so we just have to make it doable and achievable for us. And your example is an example that we have seen so many times in all of our collections just through the years. And you know, you just have to one little step at a time. So, and we can talk later, Carol. We can, you know, talk
Yeah.
forever. So,
Definitely.
yes, Piper. Yes.
When relocating vertical slide cabinets, can anyone recommend the best way to stop them from moving if not filling up individual unit trays?
What do you mean? What are you moving again?
Vertical slide cabinets.
Vertical slide cabinets, you're moving them. What's the question?
I think it's if you're physically moving the whole slide, how do you stop them from moving around?
The slides?
Yes. Oh, they're like. Oh, yes, that is exactly.
Oh, the cubes. Okay, so we have those carts. Those carts are great. And we're not moving them, so from me to Piper and Piper to me, we're just moving them inside the building, but then you ship them.
I send them to us.
You can answer in the microphone.
After I have a fully scanned cube, I reach out to the man who lifts heavy stuff for me, and he puts it on a truck, and then it gets to the place where they're stored.
Yes, and so our truck has air ride, so nothing shakes. And then they get really strapped to things, and those cubes, the trays fit really well inside the cube, so nothing shakes and moves around.
Yeah, the trays have maybe like a centimeter of wiggle room, not even.
Not even.
And then
Less than
within
that.
the trays, each has like a little metal stopper in between the sides so they can stay in place as well.
Nothing moves, yeah.
Hello again. Really interesting set. The last speaker actually raised an interesting question I was thinking about. So glass degradation appears to occur. I'm just curious if this is a decks of the Titanic problem. Is this all going to end eventually? In other words, is there a shelf life for all of this? And so eventually you guys really have to think about, you know, we're staving off the inevitable. So I don't know if that's the fact or not, so I was just curious about that.
Miriam, would you like to answer? Miriam, are you there? Oh, too bad. I mean, there's glass and glass. There's better glass and the worst glass, and glass has been taken care of better than glass that has not been taken care of as well. But yes, you know, everything has an end. But we have wonderful examples of archaeological glasses lasted a very, very long time. So the thing with slides, I have seen very, very old slides from the 1800s, and they look absolutely stunning. And with mounting media like Canada Balsam, that looks perfect. Like, so some slides are just beautiful no matter what. There's a lot of really great people that really know what they're doing. There are excellent technicians that prepare slides in a way that they're just works of art. And then there's just people that, you know, that's exactly the sound it makes. And that makes a difference. It makes a huge difference, the dedication and the time you put in the creation of each slide. The bubbles, you know, how you put that cover slip and how you do that, how much dispersion of the mounting medium outside the cover slip, the creation of those cracks afterwards and all of that. And the problem that you were talking about, Carol, the Hoyers and the, you know, all of that seeping in, and whether you put the nail polish or all the other things that I've seen ringing the slides. There are techniques, and the better you are at the step of the creation of the slide, the longer that slide will survive. And yes, you have to keep them the better that you can, but I've even seen slides in wooden boxes, extremely old slides in wooden boxes that are in amazing shape. So there's variation in a lot of things. Nothing is like, if you do this, you get that. It's never perfect, it's never linear. There are a lot of variables. So now we know a lot, and we can do a lot to prevent a lot of issues, but the creation of that slide is key to the survival of that slide. And I have to cut us off. I am so sorry. This was an amazing session. Thank you so much, everybody, for joining. And we can talk all we want about slides. Enjoy lunch, enjoy your conference, and looking forward to talking slides much, much more.